in the context in which selection acted. We need to understand it in the context of organisms. So organismal biology is very important to us because ultimately that's where our questions in biology come from. Life does stuff. That life that's doing stuff are organisms on which selection has acted over a long period of time, and we can understand biological processes by putting those questions into organisms. Okay, so now let's turn back to the level of the cell. Why do we still care about cells then if in fact a lot of organisms like ourselves have now become multicellular? What's the point of the cell? Why is that an important level? Well, there are two related answers to this question, really. The first answer is that the cell is the minimal unit of structure capable of independently performing all the activities of life. That is, the cell is the smallest unit that could be truly called living. Even in multicellular organisms like ourselves, we're still made up of individual single cells, and most of the fundamental processes that make us work are working in the context of this unit of the single cell. In fact, in large complex organisms like ourselves, there are a whole bunch of different kinds of cells, each with its own unique structure and function, each with its important role to play. But each of those roles is played out in the context of this fundamental, most important package. So, the cell is important because it is the fundamental unit of organization that can function on its own. And so as we take apart even the most complex systems, we often find ourselves bringing it down to how a cell functions. Now, the second reason that the cell is such an important level of organization, which is really related to the first, I mean, it's how the first works, has to do with this idea of packaging that I introduced earlier. By packaging, I simply mean the ability to separate this from that, to separate the inside from the outside, to separate one kind of molecule from other kinds of molecules, to separate one chemical reaction from other kinds of chemical reactions. The chemistry of life is very complex. And it's this complexity of chemistry that allows us to do all the diverse things we do, at least on that one fundamental level. And here's the point, it's the packaging provided by cells that allows this complex chemistry to occur, because different processes can be kept separated. Let's look at cells a little bit. Now, there's a little bit of background about cells. They're actually, in living things, fundamentally two different kinds of cells. Everything that's alive is made of one kind of cell or another kind of cell, and I just want to give you some background about this. We call these two types of cells prokaryotes, or prokaryotic cells, and eukaryotes, or eukaryotic cells, respectively. Now, prokaryotic cells are the things that sort of we've been talking about when we've been giving these arguments about the evolution of life on, or the origin of life on Earth. And like those protocells, prokaryotic cells are enclosed by a single membrane. This is sometimes called the cell membrane simply. It's sometimes called the plasma membrane. This plasma membrane or cell membrane is primarily composed of a particular kind of organic molecule, lipids. These are the fats that we've talked about before. And we're going to talk more about lipids and cell membranes later in the course when that becomes relevant. The only thing we need to know about this lipid cell membrane for now is that its chemical properties provide a uniquely effective barrier for separating things in an aqueous, in a watery environment, because water doesn't pass through it very well. And so it's particularly good for keeping molecules inside and outside separated. So what are prokaryote cells today? I mean, we've gone beyond the tide pools of the early Earth. Well, prokaryotic cells today are the things that we commonly refer to as bacteria. All prokaryotic cells are single-celled organisms. Sometimes they occur in large masses, large mats, for example, but even then, each prokaryotic cell is its own separate individual, single-celled individual that reproduces on its own. There are some other structures too. I mean, I don't want to run prokaryotes down. I mean, I have to point out that if you look at a prokaryote in detail, you see some other kinds of structures in there. I mean, some prokaryotes will have very long tails on them called flagella that allow them to move around. Some prokaryotes will have some infoldings. But by and large, prokaryotes represent a single package. Now, this doesn't mean that prokaryotes are somehow losers in any way. In fact, prokaryotes, in spite of this structural simplicity, are actually some of the most successful organisms that have ever existed on the planet and that exist today. Prokaryotes occupy a wider range of environments. They span a broader range of biochemistries than any other group of organisms, and they're extremely abundant. Prokaryotes are everywhere. For example, the number of a particular kind of prokaryote called E. coli, Escherichia coli, that lives in the guts of humans, for example, the number that live in a single human's gut exceeds the total number of humans that has ever lived on the planet. So it's not like prokaryotes should be ignored just because they're simple. Nonetheless, we're talking about packages, and this leads us to the other main kind of cell, the eukaryotic cell. These are structurally much, much more complex. Everything other than bacteria that lives, including anything we might want to call a plant, an animal, a fungus, is composed of eukaryotic cells. Now, there are a number of organisms, protists, that are single-celled eukaryotes. So it isn't that eukaryotic cells are necessarily multicellular. They can be single-celled. But all multicellular organisms, such as ourselves or birds or trees, all of those are composed of eukaryotic cells. Eukaryotic cells are primarily characterized by the fact that they are not only a package themselves, but they have inside them a very complex internal set of compartments, or smaller subpackages within, each separated from the different parts of the cell by its own additional lipid membrane. Now, the internal packages or compartments of a eukaryotic cell are something that we could spend a lot of time listing. We can memorize those details. But what I want to do is to draw this more broadly out to point out that there are really three main types of internal packages that we find in eukaryotic cells. Let me list those three main types for you and then go through them in turn. There are some internal packages that are specifically devoted to the problem of information storage and processing. One in particular that we'll talk about in a second. There are some of those internal packages that are specifically devoted to the problem of processing and storing energy. And then there's a third set of internal structures that are devoted to the problem of transporting and packaging biochemical products and reactions. That third set, by the way, I'll just tell you, is commonly called the endomembrane system, and we're going to talk about that last. Well, let's talk about information processing. The organelle that is, oh, and I should say, I should define that term, I'm sorry. We call these internal packages in eukaryotic cells organelles, sort of by analogy to the organs in an organism, the little pieces that function independently in a eukaryotic cell, we'll call organelles. So the organelle that is most associated with information processing in a eukaryotic cell is what we call the nucleus. In fact, it's the presence of a nucleus that gave rise to the name eukaryotic cells. The karyon, which is from the Greek root for nut, was the name given by early microscopists when they were first describing cells to a large dark element that they always found in the center of this particular kind of cell. And that large dark element is this huge piece called the nucleus. So eukaryotic means essentially having a true nucleus, and the prokaryotes are cells that don't have a nucleus. When you look at a nucleus under a microscope, a high-resolution microscope, you actually see that it's divided from the rest of the cell not by one membrane, but by a double membrane system. We call that double membrane the nuclear envelope. So whatever in this nucleus is actually quite sequestered from the rest of the cell. But not quite, because if you look even more closely, you can see that within this double membrane system, there are a number of pores, actually quite a number of pores. So it seems as though there are channels where things can get in and out of the nucleus. And that'll become very important later when we talk about protein synthesis. Now, the most important content of the nucleus, as you may guess, is the information that the cell contains. And that information is DNA. Actually, in cells, that DNA is physically packaged in long strings that are chromosomes. During most of the life of a cell, those chromosomes are just dispersed. You can't actually see them as individual strands. Yes, the chromosomes are actually dispersed. You don't see the DNA as individual strands. It's only when the cell divides that this DNA starts condensing and bunching up and becomes what we would call chromosomes visually, that we could see, you know, the little squiggly X and Y things that cell biologists might look at under a microscope. So that's what the nucleus does, is it acts as a package for DNA in the form of chromosomes. Now, we're going to come back to DNA starting in just a couple of lectures in some detail, so I'll leave that for the moment. Now, there are two kinds of organelles that are specifically devoted to the problem of processing and storing energy. And these are called mitochondria on the one hand and chloroplasts on another. We're going to come back to mitochondria and chloroplasts in detail at the beginning of the third segment of the course when we start talking about energy more specifically. But for the moment, let me just give you a bit of an overview. Mitochondria are the site where the cell actually obtains energy by breaking down carbohydrates and other organic molecules, but especially sugars. Chloroplasts are the site where a process known as photosynthesis, that we'll talk about later in the course, actually captures energy, captures it from the sun, and uses that energy to build these organic molecules like sugars, which the cell will use to store energy over the long run. All eukaryotic cells have mitochondria. All eukaryotic cells need to be able to break down things to make energy that it can use for cellular processes. Only plants and a few other green single-celled organisms actually have chloroplasts. So that one special organelle that's devoted to capturing energy is only found in some subset of eukaryotic organisms. There's an interesting other thing about mitochondria and chloroplasts is that they also have their own internal, complex internal structure, and we'll look later at how that structure is necessary for them to perform their functions. They also have their own DNA. Mitochondria and chloroplasts actually need to replicate themselves, so that when the cell divides, what happens is all of the genetic information that will be found in the nucleus gets replicated, copied, and separated into two daughter cells, but not all of the information necessary for building the mitochondria and chloroplasts. In fact, the mitochondria and chloroplasts themselves have to divide with some help from the cell, and then they just separate, sending some mitochondria over here, some mitochondria over there, as the daughter cells are created. The reason for that, interestingly, just as a little aside about the origin of life, is that it's thought, in fact, there's pretty good evidence that the place that these organelles came from was by some early cell engulfing another cell. Imagine one prokaryote engulfing the other, and then the cell that got engulfed becomes an organelle. And it's thought that independently, chloroplasts on the one hand and mitochondria on the other have arisen through that interesting evolutionary process. So the third and last major set of organelles found in eukaryotic cells is collectively known as an endomembrane system. This is very, very complicated, and I don't want to go through listing all the parts of the endomembrane system. Suffice it to say, the one piece of it that we want to name right now is called the endoplasmic reticulum. And this is just an extensive network of interconnecting, branching compartments, all bound by a single connected membrane. The endoplasmic reticulum forms sort of an internal transport and processing system for the cell. This is the system in which different components associated with different parts of biochemical processes that the cell has to do are segregated. You can think of the endomembrane system as kind of a processing and transport set of facilities that are analogous to the different rooms and vats and movable containers found in a chemical manufacturing plant. These different parts of the system all have to keep things separated and transport them at the right time to the right place, and that's what the endomembrane system does. Okay, so now we've seen how life is organized in a hierarchical fashion, and we've looked at a couple of these levels, organisms and cells, in a little more detail and asked why they're particularly important. In the next lecture, we're going to return more directly to the theme of information in biology and begin to explore in detail DNA.